In the bustling world of poultry farming, a microscopic threat often lurks unseen. This adversary is known as Colibacillosis. It is a disease that can cause significant problems for domesticated birds across the globe. The culprit behind this widespread ailment is a bacterium, a very common bacterium in fact, named or for short. While many strains have lived harmlessly within the intestines of animals and humans, certain specific types have evolved to cause disease. These pathogenic strains are the focus of our concern when we speak of Colibacillosis in our feathered companions, the chickens, turkeys and ducks that provide so much for us. The particular strains of that cause Colibacillosis in birds are known as avian pathogenic or APEC. These are not the benign inhabitants of the gut. Instead, APEC strains possess an arsenal of traits that allow them to invade the bird's body beyond the intestines. They are well equipped to cause systemic infections, spreading through the bloodstream. This makes APEC a formidable pathogen. The presence of APEC is not confined to one region or country. It is a challenge faced by poultry producers worldwide, making it an issue of global agricultural importance and continuous scientific study. When APEC invades, it often doesn't limit its attack to a single part of the bird's system. The infection can become systemic, meaning it spreads throughout the body. The bacteria can be found in the bloodstream, leading to a condition called septicemia. It can also colonize the joints, causing painful arthritis. Furthermore, the air sacs, delicate structures vital for avian respiration, are frequently affected. This widespread invasion highlights the severity of Colibacillosis. The commonality in the environment means that poultry are frequently exposed, though disease only occurs under specific circumstances, which we shall explore. It is crucial to understand that the mere presence of does not automatically signify disease. Most birds harbor as part of their normal intestinal flora. However, APEC strains are different. They carry specific virulence factors, often encoded on plasmids, which are small mobile pieces of DNA. These factors enable them to adhere to host tissues, evade the bird's immune defenses, and produce toxins. This distinction is vital for both understanding the disease and for developing effective methods of diagnosis and control. The challenge lies in identifying and managing these particularly harmful strains. Colibacillosis does not present itself as a single, uniform illness. Instead, it manifests in a multitude of forms, a spectrum of suffering for the affected birds. One of the most severe manifestations is intestinal cholesepticemia. This is where the bacteria invade the bloodstream, often originating from the gut. This form can be rapidly fatal, especially in young birds whose immune systems are still developing. The swiftness of its onset and its high mortality rate make cholesepticemia a particularly feared outcome of APEC infection within commercial poultry operations, leading to sudden and significant losses for farmers. Beyond systemic infections, Colibacillosis frequently appears as localized infections, targeting specific areas of the bird's body. In very young chicks, omphalitis or navel infection is a common problem. This occurs when APEC infects the unhealed navel shortly after hatching. Similarly, a yolk sac infection can arise where the bacteria colonize the remaining yolk material, a vital source of early nutrition. Both conditions often prove fatal for the young birds or lead to stunted growth and poor health in those that survive, representing a critical early life challenge for poultry flocks. The disease can also manifest on the skin or within the head. Cellulitis is an inflammation of the subcutaneous tissues, often seen in broiler chickens. It results in unsightly lesions and can lead to carcass condemnation during processing. Another troubling form is swollen head syndrome. This condition, as the name suggests, involves swelling of the tissues around the head, often complicated by following an initial viral infection. Enteritis, an inflammation of the intestines, can also be caused or exacerbated by pathogenic, disrupting nutrient absorption and causing diarrhea, further weakening the bird. In adult laying hens and breeding birds, Colibacillosis can take on forms that directly impact reproductive capabilities. Acute vaginitis, an inflammation of the vagina, can occur. More commonly, salpingitis, which is an infection and inflammation of the oviduct, is a serious concern. This condition prevents normal egg formation and laying. Peritonitis, an inflammation of the lining of the abdominal cavity, often accompanies salpingitis. These reproductive tract infections result in reduced egg production and fertility, causing substantial economic losses in laying and breeding flocks worldwide. Recognizing the early whispers of sickness is paramount in managing colibacillosis. In its acute septicemic form, especially in young birds between 4 and 8 weeks of age, the disease can strike with devastating speed. Often death is sudden, with few preceding signs. However, if observed closely, a brief period of illness may be noted. These birds might display anorexia, a complete loss of appetite. They become depressed and inactive, preferring to huddle rather than move. Somnolence or excessive sleepiness is another common indicator before a rapid decline. These subtle signs are urgent calls for attention. Upon post-mortem examination or necropsy of birds that succumb to acute cholisepticemia, the internal lesions may initially appear sparse. The swiftness of the disease means that extensive tissue damage might not have had time to develop fully. However, veterinarians often observe a swollen, dark-colored liver and spleen. These organs are critical components of the immune system and blood filtration, and their appearance reflects the overwhelming infection. An increase in fluid within the body cavities, such as the pericardial sac surrounding the heart or the abdominal cavity, might also be present. These findings, though subtle, point towards systemic bacterial invasion. Birds that manage to survive the initial acute septicemic phase may enter a subacute or chronic stage of colibacillosis. These individuals often become unthrifty. They fail to grow at the expected rate and appear generally unwell and weak. A hallmark of this more prolonged form of the disease is the development of fibrin or purulent lesions. This means there is an accumulation of fibrin, a clotting protein, and pus within various body cavities. These lesions are a clear sign of the bird's ongoing battle with the persistent bacterial infection, a battle that takes a heavy toll on its overall health and productivity. These fibrino-purulent lesions typically manifest as air sacculitis, an inflammation of the air sacs. They can also be seen as pericarditis, affecting the sac around the heart, or perihepatitis, an inflammation of the capsule surrounding the liver. Such conditions severely compromise respiratory and circulatory function. Less commonly, but still significantly, chronic colibacillosis can lead to arthritis with swollen and painful joints, or osteomyelitis, an infection of the bone. Lymphocyte depletion in the bursa and thymus, key immune organs, can also occur, further weakening the bird's defenses against this and other diseases. The mere presence of avian pathogenic in the environment or even within a bird does not always lead to clinical disease. Often, for infections to become clinically apparent and cause significant harm, certain triggering factors are required. APEC is frequently an opportunistic pathogen. This means it takes advantage of a bird whose defenses are already weakened or compromised. Understanding these triggers is essential for preventing outbreaks. The health of a flock is a precarious balance, easily tipped by unfavorable conditions or concurrent illnesses. Adverse environmental factors play a profoundly significant role in the onset of colibacillosis. Poor ventilation in poultry houses can lead to the buildup of harmful gases, such as ammonia. High ammonia levels irritate the respiratory tract, damaging its delicate lining and making it easier for bacteria to invade. Overcrowding creates stress and increases contact between birds, facilitating pathogen spread. Temperature extremes, either too hot or too cold, also act as stressors, weakening the bird's immune systems and rendering them more susceptible to infections that they might otherwise resist successfully. The presence of other infectious agents is another major trigger for colibacillosis. Viral respiratory infections such as Newcastle disease or infectious bronchitis are notorious for paving the way for secondary bacterial invaders like. These viruses damage the respiratory epithelium, the protective lining of the airways. This damage creates entry points for bacteria and impairs the bird's ability to clear pathogens effectively. Thus, what might start as a viral infection can quickly become complicated by a severe bacterial one, leading to more complex and serious disease outcomes for the affected flock. Similarly, primary enteric infections, those affecting the gut, can also predispose birds to colibacillosis. Pathogens that damage the intestinal lining, such as certain viruses or coccidia, can allow from the gut lumen to translocate into the bloodstream, leading to systemic infection. Even non-infectious factors like poor feed quality or sudden changes in diet can disrupt the delicate balance of the gut microbiome. This disruption can allow pathogenic strains to proliferate and cause disease. Therefore, maintaining gut health is a cornerstone of Colibacillosis prevention and overall flock well-being. Section 5. Seeking certainty, the science of diagnosis and identification. A definitive diagnosis of Colibacillosis relies heavily on meticulous laboratory work, beginning with the proper collection of samples. It is of utmost importance to use fresh carcasses for this purpose. Samples taken from birds that have been dead for too long can be misleading. This is because normal intestinal, which are usually harmless in the gut, readily invade other tissues after death, contaminating the samples. Therefore, for accurate diagnosis, only internal organs or blood collected aseptically from very recently deceased or euthanized birds are truly useful. Feces or intestinal contents are generally not suitable for diagnosing systemic Colibacillosis. The specific samples collected will depend on the suspected form of the disease. For cases of acute septicemia, where bacteria are widespread in the bloodstream, an aseptic sample of heart blood is crucial. As little as 1 milliliter of blood can be used to inoculate a broth medium, typically at a 1 to 10 dilution. The liver is another key organ to sample. A stab into the liver parenchyma after searing the capsule to sterilize its surface is the recommended method. For subacute Colibacillosis, characterized by fibrinopurulent lesions, swab samples of the exudate from the pericardial sac, air sacs, or affected joints should be carefully collected before they become sterile, which can happen if lesions are over a week old. Once appropriate samples are collected, the next step is agent identification, primarily through bacterial culture. The isolation from normally sterile sites, such as internal organs or blood from a fresh carcass, is highly significant. Preferred culture media include McConkey agar, which is both selective and differential. It inhibits gram-positive bacteria and differentiates lactose fermenters. Tryptose blood agar with 5% bovine blood can also be used. Blood samples, after dilution in brain-heart infusion broth, are streaked onto McConkey plates. Swabs from lesions can be directly streaked. These plates are then incubated aerobically for 18 to 24 hours at 37 degrees Celsius. On McConkey agar, most strains are lactose fermenters and produce characteristic hot pink colonies, typically 1 to 2 millimeters in diameter. However, it is important to note that slow lactose fermenting are frequently isolated from poultry and may not form distinctly pink colonies. Therefore, multiple colonies, both pink and non-pink if present, should be selected for further biochemical testing. A presumptive positive identification involves pink colonies on McConkey, an acid slant and acid butt reaction, often with gas production, on triple sugar iron, TSI agar, being oxidase negative and appearing as gram-negative rods under the microscope. Confirmation often involves a positive indole test and the inability to produce hydrogen sulfide. Pathogenicity testing, perhaps using chick inoculation or PCR for virulence genes, can further confirm an APEC strain. Section 6. A formidable foe. The challenge of antimicrobial resistance. One of the most significant challenges in managing Colibacillosis today is the widespread and increasing problem of antimicrobial resistance. Avian pathogenic isolates are frequently found to be resistant to multiple antibiotics that have historically been used to treat poultry diseases. Common examples include sulfur drugs and tetracyclines, which have seen extensive use in animal agriculture over many decades. This resistance diminishes the effectiveness of treatments, making it harder to control outbreaks and leading to prolonged suffering for the birds and greater economic losses for producers. The bacteria are adapting, and our old weapons are becoming blunt. The issue is not just resistance to a single antibiotic, but often multi-drug resistance. It is not uncommon for APEC isolates to be resistant to more than 3 different classes of antibiotics. Studies have shown that a high percentage, sometimes between 70 and 90% of isolates, exhibit resistance to drugs like streptomycin and gentamicin. This alarming trend means that veterinarians have fewer and fewer effective options when faced with a Colibacillosis outbreak. The selection of an appropriate antibiotic becomes a complex decision, ideally guided by specific susceptibility testing of the bacteria isolated from the affected flock. Resistance to fluoroquinolones, a class of critically important antimicrobials in human medicine, is also a growing concern, though some older studies reported higher susceptibility rates for drugs like enrofloxacin. More recently, the emergence and spread of extended-spectrum beta-lactamase, ESBL, producing in poultry populations has become a major public health worry. For instance, a study in Taiwan conducted between 2011 and 2020 found ESBL producing in a notable percentage of poultry isolates. These ESBL strains often exhibit high levels of resistance to many crucial antibiotics, including penicillins, cephalosporins, tetracycline, gentamicin, enrofloxacin, and trimethoprim sulfamethoxazole, severely limiting treatment avenues. The implications of such widespread antimicrobial resistance are profound. Firstly, it makes the treatment of Colibacillosis in poultry increasingly difficult and costly. Secondly, and perhaps more worryingly, food-producing animals can act as significant reservoirs of antimicrobial resistant bacteria and resistance genes. There is a potential for these resistant strains or their genes to be transmitted to humans, possibly through the food chain or direct contact, thereby compromising the effectiveness of antibiotics used in human medicine. This highlights the interconnectedness of animal health and human health, a concept known as One Health, and underscores the urgent need for responsible antibiotic stewardship in all sectors. Section 7. Guarding the Guardians, Strategies for Control and Management. Given the pervasive threat of colibacillosis and the growing challenge of antimicrobial resistance, effective control and management strategies are absolutely essential for safeguarding poultry health and productivity. While antibiotics have been commonly used to treat and control bacterial diseases in poultry, their extensive use has undeniably contributed to the selection and spread of resistant strains. Therefore, the poultry industry and veterinary science are increasingly focused on alternative and integrated approaches to manage colibacillosis, moving away from an over-reliance on antimicrobial drugs and towards more sustainable long-term solutions. Vaccination presents a promising avenue for prevention. Various types of vaccines are being developed and evaluated, including live attenuated vaccines which use weakened forms of APEC and innovative recombinant construct vaccines which use specific components of the bacteria to stimulate an immune response. The goal of vaccination is to enhance the bird's natural defenses against APEC, reducing the likelihood of infection or lessening the severity of disease if infection does occur. Effective vaccines could play a crucial role in reducing the incidence of colibacillosis and thereby decreasing the need for antibiotic treatments in poultry flocks. Non-antibiotic interventions are also gaining considerable attention for their potential to bolster bird health. Probiotics, which are beneficial live microorganisms, can help to establish a healthy gut microbiome, making it more difficult for pathogens like to colonize. Prebiotics, non-digestible food ingredients that promote the growth of beneficial bacteria, work in a similar fashion. Symbiotics combine both probiotics and prebiotics. Phytobiotics, which are plant-derived compounds, are also being explored for their antimicrobial properties and their ability to enhance immunity and improve gut health, offering natural alternatives to traditional drugs. Ultimately, sound management practices form the bedrock of colibacillosis control. This includes stringent hygiene protocols, especially in hatcheries, to prevent early exposure of chicks to APEC. Careful management of stocking density is crucial to avoid overcrowding and associated stress. Minimizing environmental stresses such as poor air quality or temperature fluctuations is equally important. Nutritional strategies, including the use of specific feed additives and dietary interventions, can bolster the bird's immune systems and gut integrity. Furthermore, changes in housing systems, such as the move away from conventional cages towards enriched environments, may also contribute to reduced disease incidence. Section 8. The price of disease, colibacillosis and its economic toll. Colibacillosis is not merely a health concern for individual birds, it casts a long economic shadow over the global poultry industry. This disease is recognized as one of the most economically important bacterial diseases affecting poultry production worldwide. The financial losses incurred by farmers and the wider industry are substantial, arising from a variety of direct and indirect impacts. These economic pressures drive the continuous search for better prevention and control methods, as the viability of many poultry operations can be significantly affected by the burden of this pervasive infection. The most direct economic losses stem from mortality. Colibacillosis can cause death in birds of all ages, from young chicks suffering from omphalitis or acute septicemia to adult layers succumbing to peritonitis. Even when birds survive, their productivity is often compromised. Infected broilers, destined for meat production, exhibit reduced growth rates and poor feed conversion efficiency, meaning they consume more feed but gain less weight. This directly impacts the farmer's profit margins, as feed costs are a major component of poultry production expenses. The failure to thrive means less marketable product. In laying hens and breeding stock, colibacillosis leads to significant drops in egg production. Salpingitis, the infection of the oviduct, can render hens infertile or unable to lay eggs properly. Even if eggs are produced, hatchability rates may decline due to infected embryos or poor quality eggs from sick birds. Furthermore, at processing plants, carcasses of birds affected by conditions like cellulitis, airsaculitis, or peritonitis are often partially or wholly condemned. These condemnations represent a complete loss of investment for those particular birds, adding to the overall economic damage caused by the disease. Beyond these direct losses, there are considerable costs associated with the treatment and prevention of colibacillosis. The expense of antimicrobial drugs, when they are effective, adds to production costs. Implementing enhanced biosecurity measures, diagnostic testing, and vaccination programs also requires significant financial investment. The cumulative effect of mortality, reduced productivity, condemnations and control costs makes colibacillosis a major drain on the profitability of the poultry sector. This emphasizes the critical need for ongoing research and development of cost-effective and sustainable strategies to combat this widespread and damaging disease effectively. Section 9. Horizons of Hope. The quest for future solutions. The battle against colibacillosis is far from over, and the path forward lies in dedicated research and innovation. Ongoing scientific investigation is absolutely vital if we are to gain a deeper understanding of avian pathogenic. Genomic studies are crucial in this endeavor. By sequencing and analyzing the genetic makeup of APEC strains, scientists can learn more about their evolution, how they adapt to their hosts, and the mechanisms by which they cause disease and transmit between birds. This fundamental knowledge is the bedrock upon which new and more effective control strategies will be built. To truly advance our ability to manage colibacillosis, there is a pressing need for standardization in research methodologies. When evaluating new management strategies, vaccines, or treatments, it is important that studies are comparable. This requires greater consistency in factors such as the dosage of challenge organisms used, the breed and genetic background of the birds, the timing of interventions, the overall health status of the experimental flocks, and the age of the birds at the time of study. Such standardization will allow for more robust conclusions and facilitate the translation of research findings into practical, effective field applications. The quest for future solutions also involves exploring novel control methods that can reduce our reliance on conventional antibiotics. Bacteriophage therapy, which uses viruses that specifically infect and kill bacteria, is showing considerable promise as a targeted approach against APEC. Researchers are also investigating antivirulence compounds. These substances do not kill the bacteria directly, but instead disarm them by interfering with their ability to cause disease, potentially reducing the selective pressure for resistance development. Furthermore, the development of faster, more accurate, and field-friendly diagnostic tools will enable quicker responses to outbreaks, limiting their spread and impact. Looking towards the horizon, the ultimate goal is to achieve sustainable poultry production systems where colibacillosis is effectively controlled with minimal reliance on antimicrobials. This not only protects the health and welfare of the birds themselves, but also safeguards the efficacy of antibiotics for human medicine. It is also important to remain vigilant about the potential, albeit not firmly established, foodborne link between APEC and human extraintestinal pathogenic (APEC), as some APEC strains share virulence attributes with human pathogens. The pursuit of these future solutions is a journey of hope, driven by science, for the betterment of animal agriculture and public health alike.